ขอต้อนรับเข้าสู่งานประชุมของเรานะคะต้องขอบคุณนะคะผู้สนับสนุนหลักของเรานะคะ National Vaccine Institute นะคะดร.อัจฉริยะอยู่ที่นี่นะคะตอนนี้ก็เป็น Acting Director นะคะของ NVI นะคะและขณะนี้ก็เป็นเกียรติมากนะคะที่มาร่วมงานกับเรานะคะแล้วก็วันนี้ก่อนที่จะพูดกล่าวเปิดเป็นทางการนะคะขออนุญาตทุกท่านรบกวนเวลาทุกท่านนิดนึงนะคะในเอกสารในถุงของเรานะคะเรามีเอกสารเกี่ยวกับ TBRC นะคะซึ่งก็เป็น Thailand Bioresource Research Center ซึ่งวันนี้เราได้มาตั้งบูธข้างหน้าเลยนะคะเพื่อที่จะรับสมัครสมาชิกเป็นส่วนตัวเลยนะคะเพราะ TBRC เนี่ยเป็นcollection ที่เราสามารถที่จะถ้านักวิจัยสนใจที่จะรับวัสดุชีวภาพเป็นพวกจุลินทรีย์ไปใช้งานวิจัยต่อเนี่ยก็เข้าไปก็เป็นสมาชิกก่อนแล้วก็เข้าไปดูแคตตาล็อกของ TBRC ได้เลยก็จะสะดวกสบายในการที่จะทำรีเสิร์ชต่อนะคะอันที่หนึ่งอันที่สองคือสถาบันวิจัยที่ทำเกี่ยวกับการแพทย์นะคะแล้วก็อยากจะเป็นเครือข่ายของ V Resource ของเรานะคะซึ่งก็จะมีประโยชน์เยอะแยะนะคะไม่ว่าจะเป็นเรื่องของการอย่างเช่นมาประชุมวันนี้นะคะหรือว่ามีการคอนเนคกันหรือว่าการที่จะเรียนรู้ร่วมกันในเรื่องของการทำกฎระเบียบและการทำขั้นตอนต่างๆในการจัดการกับวัสดุชีวภาพทางการแพทย์นะคะก็เชิญสมัครได้เช่นกันนะคะมีใบสมัครแล้วก็ให้น้องได้เลยเพราะว่ามันจะง่ายกว่าบางคนก็ไม่ค่อยถนัดไม่รู้เป็นอย่างดิฉันป่ะนะคะคือเข้าไปในออนไลน์แล้วก็อะไรวุ่นวายแต่ตรงนี้มันมาตรงนี้เลยอยู่ที่หน้าห้องเองเลยนะคะเพราะฉะนั้นก็ใช้โอกาสนี้ในการที่จะพิจารณาแล้วก็สมัครได้เลยนะคะขอบคุณค่ะก็ประมาณนี้นะคะประกาศขายยานิดนึงบ้างไม่ใช่ค่ะก็ขออนุญาตกล่าวเปิดเป็นทางการนะคะ Distinguished speakers and guests, ladies and gentlemen, it is my great pleasure to welcome all of you to the conference on the standardization and ethical issues for the biomedical collection. As bioresources are gaining importance in today's world of biotechnology, appropriate management of these resources become imperative, especially management of biomedical resources according to appropriate standards and ethics will promote further utilization of these resources and enhance medical research. It will also lead the way toward progress in prevention, treatment, and service in the field of medicine and public health care. For this reason, Thailand Bioresource Research Center or TBRC, which operates under National Center for Genetic Engineering and Biotechnology, or in short Biotech under NASDA and STDA, stages this conference with an aim to publicize knowledge regarding current standards and protocols of biomedical resource collection. As you know that biomedical materials include microorganisms, cell lines, tissue, DNA, and other medical samples. So long-term storage and management of these materials are very critical for the success of using them for medical research and commercial purposes, such as innovation in vaccine development. For that, we need complete and accurate information of these samples, including their history, their origin, their unique characteristics, genetic data, their features that can show the potential for further applications. Handling of these materials requires systematic registration, storage, and update all the time, which will facilitate not only the accurate use of these materials, but also the traceability and quality control of the materials. So we realize that biological samples are kept in many research centers and universities in Thailand and around the world. However, one of the problems is that each collection has its own expertise of handling the sample, not to mention its own restrictions. So this conference will hopefully bring us together to understand common standards of management and ethical issues we should be aware of. In addition, hopefully we can develop a network of biomedical resources in which members can help each other in terms of connecting and sharing information about the materials that we have. So today we are extremely fortunate to have so many outstanding experts in this event to share valuable information about the biomedical resources at this conference. Experts coming from overseas including Associate Professor Soo Yong, Dr. Hiroko Kawasaki and Dr. Jung-Sook Lee. Also, the participants in this conference come from many key institutes and organizations such as the Department of Medical Science, the Thai Red Cross, the National Vaccine Institute, the Government Pharmaceutical Organization, CDC, BioNet Asia, and several universities. So we are glad that all of you can be here and hope that you will find this conference stimulating and initiate the discussion on the effective management of biomedical collection. Lastly, we would like to thank Biotech and of course National Vaccine Institute for supporting this conference. So with that, I hope you will enjoy the conference today. Thank you very much.
Okay, can you hear me? First, I'd like to thank Lily and the TBRC for your invitation. I have been a biobanker for the large part of the last decade, for the past 10 years, and now I have shifted away from biobanking. Rather, I have remained in biobanking, but I'm no longer so much of a biobanker. I have moved to the government side as a regulator of biobanking. So this afternoon I will spend a bit more time talking about the legal and some of the ethical aspects. I think my job today is to share with you our experience in Singapore with human tissue biobanking, and so all my remarks will relate mainly to human biobanking. Now, I understand that a lot of you may be biobankers in other areas in terms of microorganisms and others. In a way, to the human tissue biobanking, to human tissue biobanker, those are not so difficult, not so complicated, because when you deal with humans, that's where the complication begins. And today I will share with you some of the difficulties, challenges we encounter with human tissue biobanking, and I also like to share with you how we dealt with them, as well as our mistakes that we have committed over the years, and perhaps that will help you as you intend to start a biobank. Now, for a start, let me just go on with the first slide, and I would say that biobanking, is this working? Yeah. Biobanking was very popular about 10 years ago. It was, it was almost like the most sexy thing in town, and every country and many universities were very keen on biobanking. So Time magazine listed biobanking as one of the 10 ideas changing the world right now. And Obama, when he was president, the White House issued this memorandum directing government departments to budget realistically to support the biomedical resources. And in the EU, we also see countries like the Netherlands and Luxembourg competing to set up hubs in biobanking to attract funding from the biopharma, from the pharmaceutical industries. Now, there's a lot of interest in biobanking in the past 10 years, but in the area of human tissue biobanking, as I will explain later in the afternoon, that the excitement has somehow sort of quietened a little bit as the problems associated with biobanking have emerged. So for institutions who are about to embark in human tissue biobanking, perhaps this is a good time to consider our experiences, our triumphs, as well as our difficulties in the past 10 years. The first question we need to consider. In my first talk, I will be more strategic and I'll focus more on very practical tips and questions that I hope you take back with you as we consider how we should design and how should we operate a biobank. But before we start about the details, the first question is, why do you want to have a biobank? Now, this is not a trivial question because depending on why you want to start a biobank, your objectives and how you run the biobank will be different. You want a biobank that meets your purpose, that is fit for purpose. And therefore, you need to know what is the purpose in the first place. And there have been situations where a lot of money goes into the development of biobank, that the biobank is developed, but it does not achieve the purpose for which the investment went in. And that was because there's a mismatch between what the biobanker wants and what the government or the funding agency wants when they start up a biobank. So you can say very simplistically, of course, we want a biobank to facilitate research, to facilitate translational and clinical research. But what kind of research do you do in your biobank? If the purpose is to raise the profile of a biobank of your institution by engaging research, and therefore I want a biobank, if your institution has no researchers in colorectal cancer, do you want to collect colorectal cancer samples? Because there's a cost with all the collection, so you have to think about it. What's all the research foresight? And if you say in general that I want to improve healthcare outcome, very altruistic, is it healthcare outcome in your country or healthcare outcome in the nation, in the global community? It may be quite different. In some countries, cancers are more important. If you want to improve healthcare outcome in Myanmar, for example, I will focus on cervical cancer. So there are differences again. So you need to think, what is it that I want to do when I set up biobank? Second, you want to set up biobank because I want to grow the biomedical industry. I want to bring money. I want to develop the economy in the biomedical industry. In which case, you need to work very closely with the biomedical industry to understand which area they are particularly interested in. For a long time, a lot of drug companies are not interested in antibiotic development. So if you focus on infectious disease, you may not get the kind of returns that you are hoping to get from the pharmaceutical industry. Right now, of course, everybody is very interested in immunotherapy. That's the sexiest thing in town right now. Everybody is looking at checkpoint inhibitors. So if you are trying to grow the biomedical industry, perhaps that's an area you need to think about, and what sort of samples should be collecting to support that kind of industry. So first of all, you need to understand why do you want a biobank. Now, there's no question that high-quality bioresources are necessary for translational and clinical research, and is seen increasingly as a vital part of the research infrastructure, has strategic and economic importance. But not all biobanks are equally good. There are biobanks and there are biobanks. There are many biobanks in the world, and even in any country there are many biobanks. Some are good, many are not so good. So how do we must have a way of assessing the biobanks to see where they are. And there are a few ways. First of all, we'll talk a little bit more details, but there's a concept of bioresource impact factor, just like journal impact factor. You know, that some journals are more highly regarded than others. Similarly, there's a bioresource impact factor, or BRIF, which has been sort of created. Their concept is to know the reputation, to track the reputation of the biobank. How many people are using it? What sort of publications do the samples go into? The other way of looking at it is the idea of certification. So the Canadians, the CTR network, has a biobank certification. What it does is that it's a checklist of what you need to have in a good biobank. And you, the biobankers, just measure. It's a checklist that you take. You measure yourself against that checklist, and then you submit it to say, yes, I meet all these. But it's self-directed. So in other words, you say that I have met all this. Nobody's checking you. So you claim you meet it and you give a certificate, but nobody really knows whether you're honest or not. It is yourself who knows where you are. Then finally, there's accreditation. Accreditation is the idea that you have a checklist, you need to measure up to it, and on top of that, after you set your measure up to it, somebody comes and inspects and see whether it is true, that to what measure do you meet the standards. So accreditation, we have the CAP, College American Pathology, CAP accreditation. We also have the developing ISO TC276. Now, as you can imagine, the most expensive is accreditation, followed by certification, and BRIF is not something that you yourself can do. It is based on the reputation over its track record. So this is the concept of a BRIF. It was published in the Nature Genetics in 2011. So really the concept is that you look at the reputation of a biobank based on the collections, who uses the samples, how you collect the samples, and whether these samples are published in top journals or not. That's sort of ideas. Let's move on. I will go into more details on each of these a little bit more later. Now the case for central biobank. Should we have one biobank for the whole country or the whole institution, or are you going to have multiple little biobanks? And for a big hospital, my hospital is not that big compared to China. We only have 1,600 beds. China has something like 5,000-6,000 beds, so they are much bigger. You can have lots of biobanks within one institution. Should you have one, or should you centralize it? Well, there are advantages and disadvantages. If you have a decentralized tissue repository, basically everyone does its own thing. The SOPs are all different, the consent forms are different. There's a problem sometimes when we try to share samples because the consent form may not meet the requirements of the project. There's limited or no funding because there's no way a institution can fund 20 different biobanks. So each one, basically, you've got money, you get money. If you have no money, then you just do whatever you can. There's limited funding. The quality of tissue may vary due to the lack of expertise because there's less funding. Operational costs will still be high. In fact, it's higher because there's no economies of scale. There's limited resources in terms of manpower, storage space. Certain expert manpower, such as pathologists, are expensive. You may not be able to afford it. There's a lack of qualified pathologists as a result, and therefore the tissue quality may be compromised. You say this is tumor, actually it's mostly fat. So these are the issues that we have with a decentralized system, unless you're a very rich decentralized system. Then you have the advantages of a centralized system, which is, of course, because there's central funding, there's uniformity in terms of rules, regulations, and funding is centralized. So therefore, generally speaking, you are able to have economies of scale, better quality, and you can be able to do more. So those are the reasons given when my former institution decided that we will have a central biobank, because there is no way the hospital can support so many biobanks and the quality of all the biobanks are not equally good. But that's a decision you have to think through and decide. Next, are you going to have a scheduled or unscheduled collection? By scheduled collection, it means to say that I will only collect the sample if there is a project, there's a demand for it, then I collect it. Now the advantage of this for scheduled collection, of course, is a better control of the inventory. You can contain costs. You collect only whatever is needed, not things which are not needed by others, and you avoid wastage. But the problem is time. There's a delay in collecting sufficient samples to meet the research needs. If you are interested in studying retinoblastoma, which is a very rare tumor, and you have a scheduled collection, you can collect for 10 years, and you won't still have enough material. On the other hand, an unscheduled collection is where you collect all samples that come through. I collect all human tumors. I collect all infectious diseases and so on. Whether or not there's a request for such samples, I collect it for future use. This will ensure there's a ready supply of samples for researchers when they need it. It is convenient for researchers. When they want it, I've got 1,000 samples. You can also advertise, I've got 1,000 colorectal cancer. And so people know how many you have and they can come to you for those samples. But the problem is that you need significant investment up front because you're collecting before they are being used. And this is rather costly and inefficient. You require a lot of storage capacity. Your biobank becomes bigger and bigger. It is not necessarily a good thing because bigger you get, more expenses are needed. And the problem is it economically viable? Next, you have to think about managing the stakeholders. And this is a bigger problem in human tissue biobanking compared to, as I said, the microbes and other organisms. Here, the stakeholders are very complex. There are lots of people involved in this, and you need to answer their particular concerns and areas of interest. For the surgeon says, what's in it for me? It is my patient and it's my tissue. No, legally it's not their tissue, but a lot of them think that it is their tissue. And they'll say, why should I bank it with me? I can keep it in my fridge. I have control. Whoever wants it must collaborate with me. I give it to you. How I know that I will be involved in every research project? The pathologist says, what's in it for me? I made the diagnosis and I harvested the sample. Without me, you don't know whether what you're collecting is fat or tumor. And therefore, if you don't get me in play, I will not give you what you want. The researcher says, I want the tissues, I want the services, but I cannot afford it. You are too expensive. But then you say you want peanuts, you get monkeys. If you want to have good quality tissue, it comes with a certain amount of investment and you need to recover the cost. For the hospital where the patient is, the hospital CEO says, look, my KPI is patient care. What I'm interested in the power is that patient comes in, they get out of the hospital with good quality treatment with the minimum cost. Research is not really my job. Then you have the funding agency. The funding agency gives the money. He says, okay, I believe in research. I give you the money because I believe tissue banking is important. Am I getting good value out of my investment? And biobanking is not cheap, as I'll explain later. So all these are the stakeholders that you need to address. And in my second talk, I will go into more detail about how you deal with each of these and the problems and the challenges we face. Next, you have to think about governance if you want to start a biobank. So first we have a tissue access committee. The more complex a biobank, the more levels of governance you may have. But at the very least, you need to have a body, a committee, a group of people that establishes the policies governing the operation of the tissue bank. This body needs to be there to make decisions. There are decisions that you make strategically across the board for the next few years, but there are also decisions you need to make day in, day out. Can I give this sample to this research group? Because the concerns may be very different. Every project has its own unique characteristics. This tissue access committee needs to take into account the stakeholders' interests. In human tissue biobanking, those groups of people we just mentioned, if you do not meet their areas of interest, if you cannot answer them, the whole biobank will collapse. And it's not just a matter of money, and it's not just a matter of power or control. And I'll explain later how Singapore tried. Singapore is like kind of, some people call us a benign dictatorship. So government controls, the government says, you do it, just do it. Even so, it doesn't always work. And I can explain to you later about one mistake we had. Then, how do you constitute the tissue access committee? I'm sharing with you a model. You don't have to follow. You need to know what are the unique characteristics of your own institution, who are the stakeholders, and then decide how do you construct your tissue access committee. I constructed my tissue access committee in thirds. 1/3 of our committee members are surgeons. 1/3 are physicians. Surgeons are the ones who are giving you the tissues primarily. Physicians are like the oncologists who control a lot of the clinical data, survivor data, they give the treatment. So they are play important role. And 1/3 are researchers. So researchers are not the only people important, certainly. In fact, you need all three. So therefore we have all these, and in addition, I have two obligate members. Obligate members means to say that no committee meeting can start without one of these two members being present. So they are very important. If one of these two members are not present, committee member, there's no quorum. So quorum, you need to have at least 50% of your members present, but in addition, one of these two members, and the obligate members are one, a lawyer, a legal officer of the hospital, and number two, the chairman of the IRB. So these are two obligate members which are important in this. By the way, in some tissue access committee, especially in Europe, where there's a need to get public support, they also have a member from the general public, a patient support group member. In Singapore, we don't have that many of these, so it's very difficult. But if you do have these patient support group, it is good idea to have one member of the patient support group in the committee as well. So next, I focus again, remember now I'm just talking very general, big level strategic approaches about collection of specimens and data. This is very important because this can make or break your biobank. You have to decide what kind of specimens do you want to collect. Do you want to collect all surgical material? Do you want to collect only cancers? Do you want to collect samples of specific diseases? And you can have a mixture of both. You might decide that I want to collect all cancers because I don't have enough resources to collect all diseases. But if there is a project on a particular autoimmune diseases, I will collect samples relating to autoimmune diseases in a scheduled collection. When there's a project, then I collect. So you can make this kind of adjustments depending on how much money you have, and basically what is the importance of each project to your institution. Then next you have to consider what kind of specimens do you want to collect. Do you want to collect blood? And if so, how are you going to collect blood? Are you going to collect blood that is spun down? You want to collect blood that is extracted into DNA and RNA? Now, if you want to do that, think about the future. There are biobanks who have collected blood samples, spun them down, extract DNA, RNA, and then store them separately. Okay? Now, these people is economies of scale because molecular extracts much less. Blood samples you need to collect a lot more, store a lot more, cost a lot more. But the problem is that you don't know how research is going to proceed in the future. So people who have done that, they use a protocol, for example, in the past, that did not extract the microRNA when they extracted the mRNA. And so when microRNA became so big, they find that these samples are not useful because they do not have a protocol that extracted the microRNA. So we always collect some whole blood and store an aliquot of whole blood because I just don't know what happened in the future. But at the same time, you will also store blood that is separated into different components, plasma, because some work will need proteins that need plasma, some will need serum only. So therefore, we will have different kinds of samples. It depends on what you have to foresee. What kind of usage do you have for them in future? Are you also going to collect other body fluids such as urine, bowels? You can collect everything under the sun. I've got people who ask me to whether we could store for them hair follicles and so on. And you laugh about it, but when I was in Oxford, there was a group that was very successful studying hair. And the reason is because you know how much money goes into hair products every year? So I think, and it's very interesting from that study that, people from Africa, some groups of Negroes, Negro people, they never cut their hair. The hair drops quite regularly, whereas other people do. So there are implications about this. It's very interesting what makes hair more curly, what makes hair less curly, more straight, and so on and so forth. So there's a lot. So we don't laugh at it. There may be requirements for you to collect various kinds of things, but you don't want to collect everything from the big toenail to the marrow because there's a lot of money involved in biobanking. Now, how do we collect samples? We need to think about consent, which I'll talk about lots about subsequently. Our staff will obtain the operating list before the surgery to know where the patients are going to be. In a big hospital, we have something like 20-30 different operating theaters. You need to know where the patient is. So the day before, we actually monitor where they are, and then our staff will be on standby in the operating theater itself to collect the samples. Because as you know, but the faster you collect the samples, the shorter ischemia time, the better is the quality. You have to decide whether the samples are going to go to liquid nitrogen, they're going to formalin, and are you going to also collect paraffin blocks? Because there's a lot of material, a lot of research can be done on paraffin embedded material. And of course, in all cases, you have to check the consents in the case notes, or some instances like our biobank, we use to collect the consent ourselves. So all those I have to consider. So here is an example what happens. Our staff will be in the operating theater itself to ensure best quality, because by the time the sample moves from the operating theater to the pathology department, easily 5 to 10 minutes depending on where located, there's ischemia time. So our staff are there in the operating theater. Here in her left hand here, she has a flask of liquid nitrogen. In the right hand here, we have got some formalin, and so we be able to provide some samples in formalin for QC purposes. Now this is where they are located. They are located here. This is a room that is literally 10 steps away from the site of the surgeon at the patient. So they're separated by a door, sliding door with a window. So they wait there. This is the room that they use to clean the equipment and so on. And they are waiting for the surgeon is informed that they are waiting for the samples. And when the surgeon is ready, raise their hand. These persons go in, collect the samples, and go back here to process the samples. Now, upon receiving the samples, you have to photograph the samples. Why? Because some of our surgeons like to photograph their handiwork. They like to see, the specimen, and they waste a lot of time because they remove their gown and then take a photograph, and the quality is not very good anyway, and they delay the specimen ischemia time. So we say to them, we have very good camera, we will take the photograph for you and we give it to you. Please don't take the photograph yourself, we will do that for you. And then we will have to describe the specimens, do a gross description, and then dissect and harvest the specimens. Now obviously, to be able to harvest and dissect specimens, you need staff who know what they are doing. So you need to have research pathologists to be able to do that. There was net freeze in liquid nitrogen, label, document the specimen. At the same time, we also obtain a cross-section slice for quality control. What do we mean by that? I have a tumor, I take out one piece of tissue, one slice, it goes into liquid nitrogen. One slice paraffin embedded, our paraffin embedded for QC to know the percentage tumor necrosis. Next slice liquid nitrogen, next slice paraffin, so that you always have a QC for all the samples you have. And when requested, I can show you this is the H&E slide of the contiguous slice. So it tells you the quality of the sample. The sample is 90% tumor or 10% tumor, we know. How much percentage necrosis, how much inflammation, we know. So you need to have that in place. So this is of course the sample operate in the sluice room, as you can see. You dissect the mandible with a tumor. You document the sample collection, and of course in this case this hepatitis B. You need to document that. We do collect Hep B samples under Singapore law, which we'll talk in the afternoon. We can collect hepatitis B infected specimens, but we cannot amplify. So those are the rules. So again, the rules may be different in different countries. On the other hand, we do not collect TB samples. Those are not allowed under our Biological Agents and Toxins Act. The samples are then placed, we put them in foil before we put it into Kravals, and then we freeze it. And then, as I said, the other sample is made into paraffin embedded material. So the corresponding sample in the paraffin block, we put in formalin so that we can process it, do H&E staining, and then we can QC the samples that we have collected. This is the biobank itself. They're taken from the flask and placed in more permanent storage. Then we of course do the documentation of the samples in our database. Now what do you do when you collect fresh samples? So these are frozen samples. The bulk of the samples you collect, the tissue bank tends to be frozen samples. That's easy. But there's also a need for collection of fresh samples for xenograft construction, for cell line. Now these cell samples need special handling. They need special handling because normally when you collect a sample that is for liquid nitrogen, the sample is not released for research until the diagnostic process is over. So from the tumor, you take a sample to be frozen for research. The rest of the specimen goes to the pathologist who assess it and makes the pathological diagnosis, report the sample. Now, if for some reason the pathologist says that I cannot make a diagnosis on the rest of the sample, I need to look at the samples you have taken, you can basically take the samples out, thaw it, and then examine it. But if you want to take the samples to be made into cell line, that sample that's taken out and made into cell line, you can never call it back. You cannot recall. So whereas for rest of the samples, all samples collected frozen are never used for research until a month or so after the diagnosis has been out. But in the case of fresh cells that has been taken for xenograft and cell line construction, you can never review them again. So therefore, your QC has to be more strict, your criteria has to be strict. The tumor must be big enough, and it must be the rest of the tissue must be representative enough so that you cannot go back, because you cannot go back to the sample. So therefore, it is requirement for fresh cell and xenograft construction, you must always have a collaborating pathologist. And the pathologist is the one who reports the specimen later on. Because that pathologist is going to take the legal responsibility that taking this sample for research will not compromise the diagnostic integrity. He must take the legal responsibility. If he is not prepared to take the legal responsibility, it won't work. So that becomes a problem later on, as I said, that when you try to run this a biobank like this and you collect fresh cells, and if all your pathologists don't want to support, they don't want to take the legal responsibility, you get stuck. Now I'm a pathologist, and I'm also the director of the biobank. So if there's any of my pathology colleagues who want to play punk and say that no, I will not support and so on, I'll tell them that, all right, anybody who doesn't report, it comes to me, and I'm the head of the department. So I know who is the one who's naughty. So anybody doesn't come to me, I will report it. So you need to have that kind. That's how at least we manage to resolve it. But it's not easy. If you have to think about how you're going to resolve this problem, you have to determine the site and the size of the sample. So normally we do not take any sample from a specimen for research unless the tumor is at least 2 cm in diameter. However, if you have, you're taking for fresh cells, you may be more stringent. You will say that I will only take it if the tumor is at least 3 cm or 4 cm in diameter. You may want to be more stringent because you cannot recall that specimen again. So this is the thing, and of course you have to make sure that our researchers, they understand that when you're asking for fresh cells, you need to prepare. It's not like frozen tissue. I just request for it. You cannot just request. You need to proactively engage the biobank and collect it prospectively. And for that reason, we have a brochure out for researchers. They know that's what you need to do to collect fresh samples, the requirements. You need to have consent before that. You cannot have it retrospective consent, meaning to say that, what do I mean? You see, for frozen tissues, we allow retrospective consent. We'll talk about it more later, but it meaning to say that you can take the frozen tissue first, don't use it for research for 1 month anyway, then take consent from the patient. If the patient says, I agree to let the samples to be used for research, then you can be used. If the patient says, no, you throw away the specimen. No harm done. You can do that if it's frozen. But for cell line and xenograft, you cannot. Because you take it, you must straight away implant it into the animal. So for that, you must always have prospective consent. Retrospective consent is not allowed. Same thing with blood. You cannot do retrospective consent. You cannot take the blood from patient and then after that, after you poke the patient says, no, I don't allow you to take blood. What do? You cannot put it back again. So that one is different. But for tumor is different. The patient has given consent for the tumor to be taken out. I've got a cancer, I want it taken out. I've given consent for it to be taken out. From the tumor that's taken out, I've taken a subsample for research. That's different. That one you can throw away. That doesn't affect the initial consent to have the tumor taken out. It's different for blood. You don't need to take this additional blood. So you cannot take it unless you've got consent. So these are the things you have to consider when you have got fresh samples. You need to be prospective consent, cannot be retrospective. You must have a pathologist. No collaboration with a pathologist, it will not work. You have to concern about the patient safety and of course RB approval, and of course there's always a cost. And this is very important, that you need to have a pathologist involved in this because you cannot recall. I can tell you a bad story about what happened many years ago in one of the hospitals in Singapore, where the surgeon took out a piece of tissue from a gastric ulcer, gastric cancer patient. Well, they suspect gastric cancer because the ulcer looks malignant. So they took a sample of it and passed it to a researcher for research without informing the pathologist. The rest of the specimen goes to the pathology department. The pathology department says, okay, I received a specimen, there's a hole in the center, probably for research, but I don't know what it is. But I look around at the rest of the sample, and the rest of the sample has no malignancy. So they reported a chronic gastric ulcer. 1 year later, the patient came back with metastatic cancer all over the body. There was cancer. If you go back to the original sample that was taken for research, thankfully had not yet been used, the cancer was present in that piece. So therefore, you have to be very careful about protecting patient safety. If the piece that you've taken out, especially if a gastric ulcer is famous, that cancer can only be in a small area of the cancer. And that can be actually the only piece of cancer available, and patient can suffer harm. So you have to think about that. Particularly with fresh tissue sample collection, criteria must be much more stringent. After the sample is collected and the rest of the specimen goes to a pathologist for reporting, with a big tumor like liver, kidney, you've got big pieces of tumor. The pathologist will sample the tumor for diagnosis. After the report goes out, there's still a lot of tumor in the specimen. It's in formalin. Don't waste it. So we go back in and collect additional tumor tissue from the specimen, make it into paraffin blocks. From these paraffin blocks, which are done purely for the purpose of research, these are not diagnostic blocks. They are not used to make the diagnosis. The diagnosis has already gone out. This is research blocks. And these can be used for a lot of research that does not require frozen tissue. Which means to say, if someone comes to me and say, I want to have a particular piece of tissue, I say, what for? For what kind of research? And we find that I want to use for IHC. I say, don't use frozen tissue. You can give this sort of samples instead. Of course, from the paraffin embedded material, you can make tissue microarrays, and from tissue microarrays, of course, you can screen with in situ hybridization, double staining with immunostaining, and in situ hybridization, and so on. So the pathology, our biobank also provides research services in constructing these tissue microarrays and providing this material for research. The blood has to spun down. What we do with the blood is that we keep aliquot of blood, whole blood, some blood, we spin it down and we separate the components and store them separately. In general, we collect blood as whole blood, plasma, cell, and then the cell components, as well as serum. So we have different components depending on how much blood can you collect, and that also depends on your patients. Generally, we ask for at least 10 mils of blood. If you are able to, we collect 20 mils of blood, but It's not possible to get anything more than that. What about clinical data? A piece of tissue is not useful unless it's accompanied by clinical data. So therefore, you need to be able to get a clinical data, but how do you get it? Now, there are two models. There are two major biobanks in Singapore. The first is the SingHealth Tissue Repository. The other is the NUH Tissue Repository with the university where I'm in. And these two models have different approaches. First, the SingHealth model. The SingHealth model will only keep basic demographic data. That means to say that we'll keep data of the diagnosis, the patient, the gender, age, and so on, but we don't have other clinical data. And so if you want to have clinical data, the researcher comes to us, they have to decide what clinical data you want. Give me a list of all the clinical data you want. I will then go to the clinician who contributed the samples. The clinician who contributed the samples is their patient, they control the data. They will then be able to search for the data that you need. And they must be, of course, a collaborator in your study. So they will collect the data that you need for your research and then to bring it to not to you directly, but comes to a biobank. The biobank will code it. So the biobank is the trusted third party. We hold the key between the patient's name and IC, identity card number, with the de-identified code. We call the TRID. So the tissues are labeled with the code. The clinical data comes to the biobank. The biobank then replaces the personal data with the code and will give the researcher the code. So therefore, the researcher gets the tissue as well as the clinical data that is linked with the code. So they get de-identified information and tissue. So this is the model. So the advantage of this model, of course, is that you only look for the clinical data when you need it. But of course, it means that every time you need it, you have to get the clinicians involved to collect the data. The other model is the NUH model. NUH model in the university, you have a whole team of staff who basically search the hospital records for the clinical data. Every time you collect a tissue, you search the clinical data and collect the data. But then, which data are you going to collect? Potentially, there can be impossible numbers of data. We collect 98 data points, including all those data you have. But I can tell you, there is no way you can have a database that's comprehensive enough. Sometimes people want data you cannot imagine, and there's no way you can have everything. We talk about whether, smoking and so on. Yes, these are standard data. But people come in and say, I want to know whether this person's daily intake of food is more than how much calories. Does this patient take sweets or not? Does this patient drink milk or not? These you cannot predict. So that's a situation becomes very difficult. Okay, but on the other hand, the NUH tissue repository model is good in the sense that you can advertise that I have got samples with the following data. If you need it, you can come to me, and therefore you can give you this kind of clinical data associated with the tissues. The bad part is that your database gets bigger and bigger and bigger, and with it, the cost goes higher and higher. Next, quality control. We'll talk about standardization in this workshop, so therefore this is an important component. For human tissues, there are two areas. First, you need to do a histology review. So I already mentioned one piece of tissue is taken out and assessed by a pathologist, which means you need a pathologist to assess the samples and get all this data. Second, nucleic acid. So a portion of tissue is assessed for nucleic acid, and basically we do electrophoresis, spectrophotometry, and looking for the double bands and the standard RIN index. So this is histology, just to show you here that you got three pieces of tissue. These are frozen. That's why the morphology is poor, because our frozen tissues here, and you can see that here these three have plenty of tissue. On the other hand, the tumor is there, but you may not be able to see the tumor because the tumor cells are these little cells here. They form less than 10% of the tumor. So obviously they are different. So depending on your research, if your research requires at least 10% tumor, you don't give them samples like this. So you need to have the QC in order to decide what sorts. And generally speaking, we do not give tissues to our researchers unless it's 30% tumor. And so, but then if the researcher says, look, I need more samples, but I can take samples with less than 10% cellularity, okay, we can give you this, at least 10%. Then, of course, this is a molecular, the standard looking for RNA integrity and looking for RIN index. What is needed for quality tissue biobank? I think that to assess whether your biobank is of good quality, there are a few things that have been done. I mentioned to you earlier on about the BRIF and certification and accreditation. There is also this tool that has been developed by ISBA. This is the Standard Pre-Analytical Code, or SPRAC. What it does is a set of questions, a checklist to ensure that you meet the minimum standards for the specimen integrity, and that is something that you can do. And on top of that, linked with this spread, they also have external quality assurance. You can send your samples to them and they can check to see the quality of your samples meet their standards. So these are things that you can do, and this is very important here. I like to show this picture because these are two specimens. These specimens was collected in 2003, and at that time, doing a Ki67, you can see it's very high Ki67. The same paraffin block in 2011, the Ki67 shows very low proliferation fraction. And always I say you can make a high-grade lymphoma, a low-grade lymphoma, by just keeping it in a tropical climate for 10 years or so. The antigenicity can degrade. This affects proteins. This also will also affect RNA. So here it's not very good because I expanded, but this is from a paper. This is from work that's done in NCI, and you basically can see that if you increase the ischemia time from 10 minutes to 20 minutes to 1 hour, you can see phosphorylation of ERK1 and 2 increases, which means to say you are interested in the ERK pathway. You might think that this tumor has activation of the ERK pathway because phosphorylated ERK1 and 2 is expressed. But that is actually the result of ischemia, has nothing to do with the primary tumor. So this is very important to make sure that your ischemia time is as low as possible. All these have got to do with QC. So this is a proficiency EQA system, and as I mentioned to you, you can subscribe to ISBA and check whether your quality of the samples is in keeping with the standards. Next, you need to have an electronic database because you cannot just do a paper if especially you have got more samples. We collect about 1,600 samples every year. You just cannot afford to have a paper system. You need an electronic system. The one we use, you can choose many vendors out there. The one we have used is something called Open Specimen or CA Tissue. This is commissioned by NCI. I have no financial links with them. I'm just sharing you what we have used, and we have used various things. We initially started off with Excel. Then from Excel, we developed our own system. And then when the specimens grew, we realized there's not enough. We got hold of a commercial system that has been commissioned by the NCI and so has international reputation. Many universities and biobanks in the world use them. It's a web-based system, and they provide everything from specimen tracking. You see there are many different modules from collections all the way down to shipping and tracking. So it helps you manage your specimens. Essentially, for a good biobank, every specimen you need to be able to tell its whole history, from the consent taking, who took the consent, what's the nature of consent, until it's actually shipped out to who and what is the MTA. So everything has to be linked throughout. You need to know who has taken it, and if it has been taken out before and put back, that is absolutely important data that you need to keep. So all these are information that you need to think of. So in the past, for example, we used to keep specimens to save space. One vial with 3 specimens of tissue in one vial. But if you take out one piece of specimen, then you put it back again, then you have to document that it has been taken out before. Although we have done experiments to show that this will not affect the integrity, even so, you need to document has taken out once before. In the end, later, we decided there's just too much paperwork and the documentation. We just put one specimen in one vial. But the problem is, you increase your storage space needed. All these are problems. There are a lot of weighing. You have to decide pros and cons. Annotation. Again, you need to be able to tell what is the nature of the specimen, the characteristics of the specimens. With microorganisms and so on, you can basically tell the molecular testing, molecular characterization. Human tissue is not just molecular characterization, it's wider than that. So first, the most precious of our specimens are the xenografts and cell lines. So for the cell lines and xenografts, we have the full history. So for every xenograft, for example, I call it a xenograph passport. The passport has all the data from how it was collected, the diagnosis, clinical history, treatment, the stage of the disease, when was it collected, relapse history, and so on and so forth. And then we have the specimen information, such as what's the nature of the consent, who was taken, who took the consent, what's our consent status, QC in terms of the percentage tumor and so on and so forth, how many vials of the normal tissue and so on, frozen tissue we have. We then do histology data, such as what's the type of tumor, the grade of the tumor, classification of the tumor, and so on and so forth. We also look at the immunophenotype. So IHC for the most common markers for that particular tumor. And this list is different for different tumor. This is gastric cancer. So because it's a xenograph, every time you passage the sample, the phenotype might change. So you need to have data for every passage, so to speak. So this is the first xenograph, which is usually after 3 to 5 passages. And this is what we have here for various markers. And already you can see there's some differences. Some markers which were originally positive can become weak positive, and some may actually change from positive to negative. So you need to actually document all that. And this, of course, with the passport, we put images. This is a primary tumor and the xenograph in terms of morphology. In general, you've done a lot of xenograph, you know that the xenograph always look more high grade because it's always the clone that's more aggressive that's selected out. Then you have the phenotype. So you can see that with some markers, what's negative before has become positive later on after a number of passages. Now you need to know that because the drug company that's going to test this drug, if it's going to target BCL2, you need to know that the primary tumor actually was negative, even though it works very well with your xenograph model. And that's a big problem because a lot of times the drug companies say that a lot of xenograph don't work. They don't work because it works very well. My drug works very well in the xenograph. In the rat, it's 100% response rate, but the patient doesn't respond. Well, this is the reason. There's a reason for it. Then you think about the equipment. Equipment is a less of a problem. It's a money problem. It's a money problem can be solved easily. Human problem is very hard. So it's about how much money you have, and you can, but the basic things, you need to have a secure facility. You need a facility with 24/7 monitoring, CCTVs, oxygen sensors, because you're dealing with liquid nitrogen. Liquid nitrogen can vaporize and can kill your staff if you do not have liquid nitrogen oxygen monitoring. You need to have training for your staff so that they know how to handle liquid nitrogen. Equipment-wise are more or less the same. You got freezers, liquid nitrogen tanks, backups, portable devices for collection, QC, and histology specimen for QC purposes, and so on. Now, I have still got time, right? Is it? I've got half an hour, right? Okay, correct. So, I don't want to speak too fast because everybody knows my student call me Machine Gun Tan because I speak too fast. The British think that I speak faster than them, so they find my English difficult to understand because I speak faster than them. Okay, so access policy. We need to think about access policy, and this is a very important component that can make or break again. Who can use your samples? And I like Dr. Obata's famous statements. He is very experienced biobanker, chair of the ANRC before, and he says that what we are talking about, what we are doing is biobanking. We are tissue. We're talking about tissue bank. It should not be a tissue vault. You know what it means? You keep tissue in the vault and you never take it out again. It's never used. That's not the purpose. A tissue bank, you need to deposit, you need to withdraw. If it's only deposit and never withdrawn, there's no point. But the problem is that there are many tissue banks that are run as if they are vaults. I like to collect. I have a huge tissue bank. My tissue bank is millions of samples. Yours is only a few thousand. And I find very proud. It's not a good thing. Nobody ever used your tissue. You should look at a tissue bank. In fact, I always say that the best tissue bank is the tissue bank with 0 samples. 1,000 samples come in, 1,000 samples go out. I mean, that's the ideal. There's a huge usage. That will never happen. But I think we have to consider who should have access. Now, who can access the tissue? What are the conditions for access? Should it be only my institution? And in ANRC, I did a little kind of a survey because there are biobanks that only allow their own institution to use, or only people in my country can use it, or can other people can use the samples as well. Are you going to share with others or not? I must tell you that if your purpose for having a biobank is high impact factor publications, you want to share with the whole world. because all the high impact factor journal papers, you looked at it, it's a whole lot of contributors from people from all over the world. So if you're going to do in one institution, you are, no matter how smart you are, it's very difficult to have very, very high impact factor publications. Then you have to think about publications. If they provide samples to you, do they have rights to the publications? What about the fee? What about IP? How are you going to share these with them? What about stakeholder interests? You consider, do you collaborate with the clinicians? Is it mandatory? In other words, if somebody wants to use the tissue, must they involve the surgeon who contributed the tissue as a collaborator as a mandatory requirement? All this has got to do with the stakeholder interest. So you have to think about it. How are you going to factor the tissue bank in the policies in terms of access policies? Of course, IRB and RCA's issues, you need to have all those documentations in place. What sort of documentation in place? Is the MTA good enough? Must you have an RCA? And in RCA, must you consider IP issues? All these have to be considered in terms of your tissue access policy. Now, next, on top of publicity, I hope at the end I will have some question or answer, so I'm trying to summarize a little bit here. But publicity is very important. It is something that many tissue banks do not put enough emphasis on. And because we always think of ourselves as scientists, scientists don't really like publicity. We want publications, not newspaper articles about us. But if you are running a tissue bank, you realize that you are actually managing a public resource. And therefore, it is very important to satisfy all your stakeholders, including the general public. And so publicity is important in order that people are happy to contribute samples to you. So publicity is needed, of course, for your researchers who will be able to find you and get tissues from you. So, of course, you need to have a website, essential. Everybody should have that. If you haven't got one, you must have one. And your website must be useful to provide enough information of how you can access samples, what kind of samples you have, and how you basically get the details of the samples, and so on and so forth. The Singapore Tissue Repository, these are the two websites. This is the NUH one, which is a smaller repository. This is the Singapore Tissue Repository, which is a bigger repository. And here you can basically get all these materials there. Next, you want to look at brochures. Now, this is particularly important when you take consent, because for the patient, this may be the first time they learn about biobanking, and the first time they hear about biobank, and you want them to give tissues to you. So you need to educate them. So the tissue consent procedure, we'll talk about more later, actually is a process and takes time. But in addition, sometimes people cannot decide there and then. They need time to absorb. They need time to read. So we produce these brochures, which we give to the patients. We encourage them to bring it share it with their relatives, for their wives, their spouses, have a discussion about what they're about to do. So we need to produce that. We need to engage the public. And therefore, the tissue bank director has to go to the reporters. So I go out to reporters, and go to newspapers, and so that the public is aware of how tissue banking helps patients in general. And you need to get buy-in from the public to support biobanking. Of course, this is also important to convince your governments and your funding agencies that you're doing work, so that's and that is worth supporting. So that is also important. Now then you have to balance demand and supply, and this is about long-term sustainability. Now give you an example. This is a short period, but just tell you the experience of a biobank. Biobanks always grow in size, just like men after middle age. This is a problem. You get bigger and bigger, and you need to consider the cost. Within a period of 4 years or so, from 2007 to 2011, the specimen numbers doubled. And if we look at the freezer occupancy rate in our labs, it's just increasing. More and more freezers, you bring a new freezer, it gets full again and again. How do you sustain it? Now, statistics show that only about 10% of samples collected in a standard tissue bank are actually used for research. And that is a statistic you must keep in mind. 10%. No hospital will operate if only 10% of your beds are occupied. No hotels will remain standing if only 10% of the rooms are used. And yet we are operating a system where only 10% of our samples are used. How can it be sustainable? So this is something we have to think about. And the cost of biobanking is not liquid nitrogen. That is the least and the cheapest of all. It is not the cost of liquid nitrogen, it's a cost of collection, the manpower cost, the cost of collection, annotation, the processing, the storage, your utility costs every year. You've got minus 80 freezer, you know how much energy it takes up, and especially in a tropical climate like where we are in, it's a lot of energy that's been taken up. So all these are things that you have to keep in mind. Revenue recovery. No problem, charge from the researchers for the use of the samples. You cannot. If you charge the full cost, nobody will be able to afford it, and therefore your biobank will also collapse. The reason is very simple. If only 10% of the samples are used for research, are you going to charge that researcher the cost of the other 90% of samples that you've collected and not used? So you have a problem here. How are you going to recover the cost in full just from the researcher? That's the challenge. The cost of a medium-sized tissue bank in Singapore is about $1 million a year. Now you can say that Singapore is very expensive. Yes, it is expensive. But even in Thailand, I don't think it's going to be cheap. So you have to consider the cost and what is the long-term sustainability. One of the big errors of many biobanks is that there's a lot of enthusiasm and a lot of plans when you start a biobank. But when you start a biobank, there's never a thought about how you're going to close the biobank. You remember that biobanks have a very real chance of closing down. With only 10% of samples being collected, it's a surprise that many are standing. Many biobanks close within 5 to 10 years, which means to say that you also have to think as you start a biobank, if the inevitable happens, what are you going to do with the samples that you collected? How are you going to manage it in a way that is ethically, legally, and socially socially acceptable? How are you going to dispose of that samples? And that's something people don't think about. But I think if you're going to start a biobank, you'll think about that. Can you afford to collect everything? My answer is always no. You have to be very careful about what you want to collect. So I will stop now for the first talk, and maybe I'll spend some time for some question and answers before you forget your question. I do know at the end of the day we have another longer Q&A session. Happy to answer any questions you have. Thank you.
Thank you very much, Associate Professor Soo Yong, for your very informative talks, and from your expertise in both as a pathologist and a head of the biobank. So we learned a lot about the policy and considerations and limitations to share.
So far, this is just an overview.
Oh, sorry.
My name is Suwani from TBRC. I'm a curator of a biobank, microorganism collection, but not the human specimen. I wonder, like, what are the data standard you follow? Like, you have different type of tissues, and each tissue, there are different kind of data you have to collect. Like, what are the minimum data that you need to collect? What kind of standard you follow?
Okay, so there are standards relating to the specimen viability. Those are easy. So as I've shown earlier on, consent, type of consent, nature of consent, whether conditional consent, not or absolute consent, universal consent. Then in terms of the specimens, time of collection, ischemia times, all those in terms of the QC, percentage tumor, inflammation, all those are standard. So that is the specimens information that we collect for all, regardless of the type of specimen. I think your question relates to different tumor types. What sort of additional data we collect specifically with PDR1 being so important? Every sample we look at PDR1, but the problem is that the standard for assessment of PDR1 is different. Lung cancer, you basically look at the percentage tumor, the DPS. in the case of the gastric cancer, you look at the percentage of PD-L1 staining in the tumor cells as well as inflammatory cells. So every tumor type, the standard criteria will be different. So I can't give you a whole list. It depends on the tumor type that we collect. So you need to have a pathologist involved, and sometimes you also need to have an oncologist. That's why the tissue audit committee is the part that provides advice as to what sort of data we should be collecting, because the science in this field is moving in this direction. We need to collect this sort of data to support the research in this area. So it varies for every single tumor.
Thank you very much.
There's a question.
Very much informative presentation. I came from Japan. I'm a worker in the microbial biobanking. So in the last slide, I have same question, same problem. The same with the human biobanking. That we collected so many samples, strains, but very small number of percentage are used by the outside the users. In the last slide, how do you think we should? Of course, we should do. In the finally, you explained to many way, but in the first, also that in Japan, the human biobank has same problem. So it take much cost to take here, but they should do.
Okay.
Please tell.
What will you? But my decision was, I use a figure of 3 times. If I have, let's say, I look at the usage of colorectal cancer. Let's take colorectal cancer. I look at the usage of colorectal cancer in the past 5 years. So what is the total number of colorectal cancer issued out in the past 5 years? Let's say it's 100 samples. I multiply that by 300 samples. If I have got 300 samples in a biobank, I'll stop collecting. I want to make sure I have at least enough specimens 3 times more than the total collection in the total samples that has been drawn in the past 5 years. And if I exceed that number, I stop. I don't collect anymore. I stop collection. So in microbial may be a bit different because you have to characterize the microorganisms, and in a way then you see that if this microorganism is very similar in terms of genetics or whatever to what you already have, and you've got a lot of them, you don't need to collect so much. I mean, I'm not going to tell you how to do your job, but in microorganisms you have an advantage that you can always culture. We can't. You have this big advantage. So if you can culture, there is no need for you to keep so much. We have a problem because ours, once you've given out, that's gone. So therefore I need to keep enough reserve, but yet on the other hand, I cannot be keeping more and more. So I use a criteria of 3 times of the last 5 years withdrawal in my biobank. So I monitor that. So for example, over the next few years, things change. Lots of people want to do colorectal research. Instead of 100, it's now 1,000 being withdrawn in the last 5 years. Then I need to have 3,000 in the biobank. So this is what I do to make sure that I have enough tissues in the biobank, but I will not collect. So actually I say I will stop collection of colorectal cancer. So this is something what we do. Yeah.
University, and I am a pathologist too. So
Good to meet you. Very good.
And I have started to collect biobanking 5 years ago, but I only got 2 or 3 colleagues in my department for biobanking work. And do you have any suggestion or recommendation? How did you manage to facilitate or incorporate biobanking work into the routine pathology work?
Yeah. First, very good to meet you, fellow pathologists. I hope there are more people. So the reason is because he wants me to develop robotics and AI. So this is what we are doing. Answer to your question is that we use pathologists from neighboring countries. Because in Singapore, to work as a pathologist in Singapore, you need to have qualifications from either 3 places: either the Royal College of Pathologists of UK, the Royal College of Pathologists of Australasia and Australia, or the American Board Certification plus 2 years of fellowship in a recognized center. So not many people meet those criteria, particularly those from the neighboring countries like Myanmar and Philippines. These are the 2 countries from whom I recruit our pathology associates. in a way they function as research pathologists. They have all gone through at least 4 years of pathology training in their own country. They are passed as pathologists in their own country. They know enough of gross pathology. You can tell between tumor and benign, what tumor and what's not tumor. Their qualifications do not allow them to practice as pathologists in Singapore to sign out patient reports, but they can function as pathology associates. And because of the salary difference between Singapore and these countries, we are able to hire them to Singapore to function as pathology associates. So that's how we're able to get hold of them. And many of these are very good. We train them, and some of them are very good. One of my pathology associate is so good. I mean, I'm a lymphoma pathologist, as you might know, and I trained him, and he's so good that for a moment he got himself hired by a private hospital, by a private pathologist who got him to screen the cases, read the lymphoma, prepare the report, and the Singapore pathologist look at the slide and then sign it off. That's a misuse of him. But what I mean is that these persons can be very, very good if you provide them with the right training, and they're able to do these things which I want, the QC as well as the harvesting of the samples. And this is what we do. Getting our own pathologists to do it is very difficult, practically impossible. Our residents do help in biobanking to harvest our samples. We pay them a certain sum of money every sample that they harvest, because this is on top of their job. So we pay them a certain sum of money for harvesting samples for us, and they can use that money to buy books and so on and so forth. So that's the other mechanism that we put in place. Does that answer your question?
Yeah, but it's about the extra staff and money.
It's about extra staff and money. I told you from the start, biobanking is not cheap. If you really want to do biobanking well, it is an investment. So remember my very first slide. Very first slide is the most important slide. Are you sure you want to have a biobank? Why do you want to have a biobank? You must answer that question. And if you are convinced and buying that this is important, we want to go it, you really need to have that kind of affirmative action before you go. Because once you start, you will find that all these things are very challenging. Any other questions?
Any other questions? Comments? If not,
Thank you very much for your excellent presentation. I have 3 questions. The first, how many biobanks for healthcare in Singapore? And the second, do you need to use like a law enforcement or not? And the third question is about the mutual benefit, or how to share the benefit.
Very good questions indeed. Biobanks. The government in Singapore has decided that Singapore, a couple of years ago, they divided Singapore into 2 halves, West and East, and with 2 big tertiary centers that are able to do the top level of most complex procedures, such as transplants. So transplants are only done in these 2 major hospitals. West, we have the National University Hospital where I work in. The East, we have Sing Health, where I used to work in. So it's very small. So these are the 2. These are 2 big ones. However, there are multiple small biobanks that we do not know of. Because every surgeon can keep a specimen in his own fridge. He is also technically a biobank, but nobody knows. And this is a problem for regulation for all of biobanks. And that is going to be stopped to some extent, because now we have a new law, which I will talk about this afternoon, where I'm involved in, called the Human Biomedical Research Act. The Human Biomedical Research Act has 1 module purely dedicated to tissue banking. And the tissue framework of the Human Biomedical Research Act requires every biobank to be notified to the Ministry of Health. And they have definition about what is a biobank. And a lot of people don't realize that there are biobanks. Under this law, they are now all biobanks, and therefore they are required to notify the government. So we can answer this question in future. Right now, we still do not know, because this act is going to be fully implemented. It's being implemented in phase. The first phase was last year. The last phase will be implemented in the middle of this year, and that will cover biobank. So that will cover them. Then by then, we will then know how many. Second question about law enforcement. Do you need law enforcement? The answer is, if you have a law, then you can enforce. So the first question to the question is really, do you have any laws that govern biobanking? Or perhaps the question is, does the public feel that there's a need to have laws that govern biobanking? If you have done such a law, then you need to enforce, absolutely. So again, this afternoon, I'll talk about the Human Biomedical Research Act in Singapore, which is the first law we have that goes right down to the level of biobanking. Before that, we only have a single clause under the Human Organ Transplantation Act that says you cannot buy or sell blood or tissues. That's just very single line. But now we have a lot more, and with this law, we will then have to enforce. For the past 3 years, I have been working with the Ministry of Health, helping them to draft this law under the Human Biomedical Research Act. From middle of this year onwards, I will be transferred from the legislation and licensing to a different department called enforcement, where I'll be in the enforcement arm of the Ministry of Health to enforce that law. So that's the second bit. And the last bit is, how do we ensure mutual benefit? Well, that is got to do with the policy right from the start. As I mentioned, you need to consider who are the stakeholders, and you also need to consider why are you setting up a biobank, okay, to advance research. Advance research where? In your country only? For the whole nation? For the whole global community? You have to answer many of those questions in your mind as to why you want to do it. Are you keen to have this for mutual benefit of everybody or not? Or is for your own benefit? Some surgeons collect samples only for their own benefit. So you have to think about that. But answer to that is yes. I mean, we all know about OECD rules, and so the whole idea is that we want to go for mutual benefit. Microorganisms, very clear about mutual benefit. Human tissue, quite different. That one is harder to apply. How do we ensure mutual benefits? We, in the policy, as I'll explain later on, we have policies in place to ensure that the biobanking will benefit the surgeons, the contributors, the patients, the pathologists. Everybody must benefit. The other bit is, of course, about the reputation of your biobank. You heard about BRIF. The reputation of the biobank goes up if you share your samples with other biobanks. If your samples are used by other researchers in their publications, your reputation goes up. This is a way to encourage mutual benefit, so to speak. But as you realize, human tissues are much more complex than microbes because a lot of people feel they are very sensitive. A lot of people feel a lot of possession. They're very possessive about tissues. So trying to get mutual benefit requires you to answer their various interests before you can get people to agree to benefit each other. So, people can be selfish, but I think if you give the right message, people will be willing to share. We'll talk more about that this afternoon. Okay. Right.
Thank you so much. Very detailed introduction. So, good morning, everyone. Can I sit? Yes. It's very honored to be here and talk our activity. I came from microbiological field. In the coffee break, I had that all of the participants came from the medical field and the pharmaceutical some department in universities mainly. So and then, sorry, please. This time is please make relax. Maybe in a quite different field. And then also the Professor Dan said, microbiology is easy. Yeah, of course, easy compared with human cells. I think I know because recently also I will work together with, sometimes together with the human biobanking. So and then the problem is quite different. So I show the very simplified biobanking. Maybe you can get some imagine to use in your biobanking or something, right? I hope that. So, sorry, how to do? Yeah, I came from the National Institute of Technology and Evaluation. Our institute is under the Ministry of Economy, Trade and Industry. It's not say with the science and technology and the medical field. Just that we should focus to industry first. I should say in the first step. It means that the purpose of the biobanking is for the industry and for economy of the Japanese company something. But we are the public. I'm a public officer because this is a government institute. This is the biggest culture collection, microbial culture collection in Japan. I talk this kind of story. Just I briefly introduction of organizational structure of our institute and our collection, our BRC. And next, just also simple activity of NBRC culture collection. And next time, I'm thinking about the BRC and the standardization. And next, introduction of international standard related to biobanking. Already Professor Tan explained a little bit, but just I show a little more. And finally, responding to new international accreditation standard in Japanese biobanks. So this is a, yeah. Okay. This is, I just showed our institute. Very funny, I'm working biological resource centers, but in our institute, so many quite different centers are together. The keyword of our institute is evaluation. So that is the same thing, because the chemical management center, we evaluate how safety, the new chemicals, new products, and give the inform to the public and the company and the researchers. And this one is consumer product safety centers. How safety the product, for example, some electric product and other things. And this one is in international accreditation Japan. That is, yes, this is oh sorry. Yes, this is, we have the government accreditation body. And finally, quite a new one is the yellow one here. Is recently we should reduce to use the fuel, so and then we use the natural electricity or something. So and then, for example, the car also change from engine gasoline to the electric car. So like that, in the battery, the size of battery is bigger and bigger. So but very dangerous for us. So and then we evaluate the safety of big battery system. Such kind of facility we have. Next. And just I focus to our center, biological resource centers. We have the main mainly two places. One is Tokyo. It's a headquarters is in Tokyo, but laboratory is located in the a little bit countryside, Chiba Prefecture, the opposite of the Tokyo Bay, and there is 3 buildings. One is middle one, the culture biobanking for not the middle one, upper one. Upper one is the microbial biobanking facility. Second one is only one patent deposit center in Japan we have, and the last one is the long-term preservation center. We commercialize to the not commercialize. We support to the company to keep the very important microbes. And until now, we just start also tissue culture, but not yet. Because the Professor Tang explained to that the number of human specimen is increasing. So that maybe this facility also we are thinking to collaborate with that. Such kind of researcher and organizations, but not yet. And second, I talk about activity. This is a very simple activity of our culture collection. Culture collection is very simple: accept, preserve, maintenance, and distribute. But we don't do only this one. Here, the culture collection work with also international cooperation. Also, we have long-time cooperation with biotech also, and the genome analysis. But recently, in Japan, the company can help to sequence and no need to buy ourselves. Just how to use the whole genome sequence. We support to the researcher and the companies. And biosafety and inspection. Also, we should show the main information about biosafety, focusing to microorganisms. But no time to talk today, and I skip. And patent depository center. And the new one is industrial support, because our institute is under the Ministry of Economy, Trade, and Industry. So, and we have collaborated with company right now. The total budget is this is almost, we are the government institute, so totally I showed this budget. the industries is of course including the collaboration competitive project funding from the government or company, but almost this income came from the when we distribute the microbes and the income, the money, like that. And also patent depository centers, completely we manage from the customers. So, but other one is almost came from government support. And the number of employment is totally 150, including technicians. We manage everything. And I said the biggest collection in Japan, the number of collection is biggest. And recently more than 90,000 microbes we preserved, we maintain. And but I think we make two purposes. One is for much more standardized one, and another one is that just only screening purposes. It means that I cannot say no need to standard, but no need to quality control. I don't want to say, but I think this one is much more standardized one. So the 30,000 story and the other one is 60,000 stories. And no need to just briefly the whole like school. The microbes is effective to food industry by remediation and also biofuel and using genetic recombination in the medical field. Other things, so many things. This is information about the number of collection. In the first, the number of filamentous fungi is much more than a percentage than 10 years ago. But recently, the number of bacteria is increasing. So in the one third is filamentous fungi and bacteria, and next is actinomyces, right there. And also we have the human cDNA clones and also human gateway entry clone like that. Okay. This is a number of culture collection. How many percent you use? So almost our customer is domestic customers, industry, academy, and then very small number of foreign industries, foreign researchers. Totally is 7,000, more than 7,000 points. This one we calculated. Just I want to say that 2002, we calculate 2002 because before that, our collection is not so old. We started 2002. Before the former collection, original one was maintained in the company pharmaceutical company, Institute of Fermentation Osaka, maybe you know or not. That one's completely supported by Takeda Pharmaceutical Company. More than 15 years ago, the government, our ministry, think that in Japan, we need the biobank, microbial biobank for industry. So, and then the government decided to all of the microbes receive to the government and start to our culture collection, our biobank. And then increase number of distribution become increase. But recently the average number of distribution per year is 8,000 strain, and the number of order is 2,600 order. So and then yes, the 2 times in a week we send to the users, and some of selling office is very busy, packing and checking and other things is more than almost 4% are doing the yes, like how to say that. Yeah, Amazon, not Amazon, but I hope to be Amazon. So the third one I talk BRC and the standardization because today's topic is that, and I choose this one. This is our simple work of BRC management. This is a little bit different with human biobanking. We collect from nature, right? So, and also we do identification first, to register the collection. identification technique is so many techniques. We use molecular and morphology and chemical analysis, everything, and we select the good one. Good one is the important one. That is just our feeling, but I think in future maybe the microbes. This microbe is, for example, it's good and new and very important, so that we selected one and preserve in the freezer or more convenience way that we use a very suitable long preservation way and register to our biobank culture collection and make long-term preservation and very easy transfer the samples. We use rigid dried samples. And also in parallel, we do much more some additional data independent of microbes, and also we put the data in the database and publications. That is a very simple way in the microbial culture collection, biobanking. But I think that I should say the standardization, in which way we should standardize. It's after we register to the culture collection, we need more maintain, manage in, I'm sorry. Okay, I quit. It's okay. Yes, I point out. The biobank and the standardization, especially microorganisms. Standardization of BRC management for quality control by resource and improvement of services. Now is we manage under the ISO 9001, the quality management system. The 9001 is a standard that set up the requirement for the quality management system. It helps business and organization to be more efficient and improve customer satisfaction. And quite recently, the new accreditation document was published. The number is 200387. It's a much more focused to the biobanking, the general requirement for biobank. The document specifies general requirement for the competence, impartiality, and the consistent operation of biobanks, including quality control requirement to ensure biological material and data collections of appropriate quality. But now is until now that we are doing that one, but I'm not sure that in Japan, of course, there is not yet set up to accreditation of this one. Maybe in the world nobody do that, just only publish this standardization. But maybe in future, at 5 years, 10 years ago, maybe in the future how the come is I don't know that. And last one is providing standard microorganisms test strain to support. I think the NBRC should think that the standardization management and also providing standard microorganisms, that is a different way. So we focus to standard operation processes for microbial BRC under ISO 9001. Everything, the most important thing is the acceptance, the who, when, and where, and the data is enough or something like that. We should be careful, and of course we make a method in detail, and we should record everything. After that, microbial preservation, we should select which one is the best way for long preservation, and also QC of product. We receive the sample, but the microorganisms are different with tissue samples. We can make reproducts, but the tissue sample it cannot. So that is problem. We can increase the number of cells, but sometimes human error is come easily. So and then we should check the purity, identity, and identity with deposited original one. Using several techniques. Also, we make a procedure to check quality control. And after everything is okay, or after that, we also make a standardization or processing of microbiology for transfer to and shipping to the customers. It was all of the system was make our own strategy and own protocol and under the ISO 9001. It's a some of our facility, this is we make ampoules. They also we make procedure. The several technicians make 3 days in a week. They make more than 300 ampoules, like that. And then the number of freezer I cannot calculate in my building. Maybe nearly 60 or 70 big freezer are there, and this one is also more than 60 liquid nitrogen tank are there. Very big, like a building is refrigerator, I call. And next, I talk about providing standard microorganisms for testing. It's important in microbiological resource centers. This is our home pages. In the home pages, I showed ISO, GIST, test strain. It's a group of microbial strain authorized by ISO and GIST, and this one to be used in officially standardized examination are included in our collection as listed in table 1 or something like that. That you can see the down. It means that we have nearly 90,000 strains, but some of very small number of 90,000 one is a very important for standardized method, which like this ISO 846 or something. We showed Aspergillus niger. Of course, ATCC number was mentioned in the document, but also our collection strain were mentioned in the document. So, and the company and the researcher use this one for testing this ISO standard, like that. The totally we have it's not cover everything, but ISO we have such kind of testing in this document. Our strain was mentioned. They should use our strains, or we recommend to use our strains. And also we have just original Japanese industrial standard there. About more than 12 or I don't know 12 documents. Also in the 12 documents, our strain are mentioned to use to standardize one. And also the other pharmaceutical testing, also our strain was mentioned. Yeah, it means you can imagine that this is a document, technical standard document. So, and if our strain is not good, it's very low quality, so that the result of the test is not good. So, and then we need high quality microbes in the strain level. And other side, just I put before during the coffee break, because the professor explained that the biobanking, one of the biobanking importance is that not only keep some researcher, the user, customer can use our bioresource. So, and then in 1 year ago, we revised the idea, because the microbial culture collection history is more than 130 years. So just like that, you can imagine that. So the professor keep microbes, and some researcher want to use the professor's one to go to the laboratory and please give me this strain, and then or please look and teach me. Oh, I'm busy or something like that. But we ask many things, and then the user can use the reference one. That is a very, very long history in microbial culture collection like that. So, but after the 100 year, we think that, the culture collection is services organization. Services, yes, should be a service, and we should think about the service mind. So, and then first we make, this strain can be used for only research. This one can be used for commercialize. Just only that we do. But recently, their environment are quite different. For example, related to CBD, and also some user want to looking for the collaborator for researcher in near future or something like that. So, and then we change a little bit the mind. When we receive the microbes, first we should decide more detailed contract with depositors. Of course, some depositors, some researcher, I can give to culture collection and you can use by yourself and free. Some researcher, I can give this, I can deposit the strain to NBRC, but only please use only non-commercial, only research. Or sometimes, okay, I can deposit to your collection, but I want to looking for the collaborator. So that please mention right now, for example, the condition for utilization specified by depositor within the range of NBRC can agree. We put that so that maybe the depositor can write more detail, so that user is much easy to understand how we can use until what yes, the user also it's a user friendly one. Yes, next one. Sorry, the time is. Okay. Very sorry. It's a relaxing time because different field. Okay. Just this is I want to skip. Maybe nobody knows. So because in Japan also there is okay, yes, committee, yes, and for ISO the professor that explained about that. It's okay. I want to skip. Yeah. Okay. Just okay. Here. Yes. ISO TC 276. This one. Japanese. Standardization is the field of biotechnology processes that include the following topics. This is a normal one, terms and definition, biobanks and bioresources, analytical method, bioprocessing, data processing, right up. This one is you can go to the website and there is one. There is Technical Committee 276. Maybe more than 6 years, nearly 6 years ago was established this task. Technical committee. 5 years ago, I joined the member as an expert from Japan because only me is a microbiologist. The other one is human biobanking because the human biobank much more careful, scared about the how international standard will come. So, and then I think I joined. I explained about this one. Yes, I said the typical one. TC276 biotechnology, we work closely with related committee in order to identify standards that need the gap. This is okay, very simple. Yes, this is just briefly scope of 276. And in the 276, also Thailand is a P members. So and then I met yes, Dr. Lee and Spaldi in the meeting. Also, yes, right there. Here. And just I select the focusing to the working group 2. Working group 2 is focusing to biobanks and bioresources. And I said just quite recently, I said already published one, the 203A7 is biobanking general requirement for biobanking. And this one related, I selected the related one. This one is that general requirements for the validation, the verification processing method in biobanks. This is a general requirement, but how to validate, how to verification is this is a document for that. And the next one is that this document is very general one because including human, microbes, plant, animal, everything. So that this one the implementation guide for ISO 20387. And the last one is related to databases for microbial biobanking. Here, it's a total structure of the working group 2. It's a quality management standard over there. This is a quite general ISO 9001. The user requests to human specimen biobank, animal biobank, plant biobank, microbial biobank. This one is the general accreditation standard. It's yes, I already published one. To understand this one, that there is we are discussing a technical report 227858, the guidance for this one, and this one is verification document, and under this one, the much more specified technical standard are proposed and for the human specimen biobanks and also animal plant. The number is this one, 203A838, this one, blah, blah, blah. Yes, this one. And quite recently, the microbial biobanks, new technical standard will be proposed maybe on this June, something like that. that this is a total structure for maintain the standardization of biobanking standard. Yeah. I'm sorry, I showed that you can also see the information, the ISO homepage is 20387. Here. I talked here, including human, animal, fungus, and plant, and microorganism. But this is important for research and development, because the other ISO standard were there for clinical field, so and also food production field. So this is just only for the research and development biobanking standard. Yes, finally, just I said, maybe I'm not sure how the Japan thinking to update to the new one. Yes, biobanking in Japan, there is human biobank, mammalian biobank, plant biobank, and microbial biobank. This is one of this one is TBRC. And also in Japan, there is some project basis network. It was supported by a Ministry of Science and Technology. And in there, the very small collection, small biobank, but very interesting small collection were there. So the network are supported by Ministry of Science and Technology. And also human biobank. Recently, the Ministry of Health, Health, I don't know the exact name, Ministry of Health or something is supported to human biobanks recently. In Japan, the number of this one is much more than these banks. And this is only the Japanese website. Quite recently, the Japan Agency of Medical Research and Development. This organization also established less than 5 years ago. Maybe everybody knows in Japan there is no such kind of things. And quite recently, government think about that. The Ministry of Industry, Ministry of Science and Ministry of everything should gather for focusing to medical. So and then the agency was there. The agency think biobanking is very important. So and the first, how they did. First they make a list of all of their biobanks, human biobank in Japan. Here and then make a very simplified databases. One is this is a sample kind of samples or DNA, blood, specimen, cell. You can choose this one so that the list is there is a name of the biobanks. This is, for example, the first one, the Biobank Japan, BBJ in Tokyo University. The sample, they explain about sample type very simply. For example, DNA and something like that, and the number of samples, and the disease name, and the regulation, and how to contact, how to get the sample like that. We can quick and go to this one. First, set up this one. And recently, the government gave much money to support to biobank, to the human biobank. Yes, that is all. So, and then I think, biobank, sorry, it's okay, time is come. Conclusion. I believe that high quality biological resources support high quality research, and as a result, support human health and productive life. I think an international standardized, and no need to get the standardization, but their arguments affect the provision of high quality biological resources in biobanking. I believe that. So, and then I also want to discuss with you, but even if the fields are different, but I want to discuss and I enjoy today. Thank you very much for it.
Thank you very much, Dr. Hiroko Kawasaki, for your no, it's okay. We still have time. Don't worry. So I want to, if the audience have questions or comments, will be welcome.
Need for standardization is there because we want to share samples. If you do not want to share samples, you're going to collect samples just for your own use. Really, the need for standardization is not so strong. So you have done, in Japan, of course, it's very good in terms of standardization. But one of the observations I have is that, like the website you mentioned, everything's in Japanese.
Yes.
And does it indicate to me that at least in the area of human biobanking, there was really not much intention or desire to share samples across Japan, outside of Japan?
Yeah, of course, I think so. I think standardizing is most important for standardization is for yes, share with overseas. I believe that, but I think I'm not government person in this field. But just I want to say that last year they list up the databases first, because I think I want to say it's also difficulties because the hospital has own biobanks. They don't like to share. They don't like to show. So, but the government organized. Okay. So, and if I think that I was invited the meeting sometimes. So, and if the small biobanks also gather in there so that they can get benefit. If so that they will show the information to the government and share with everybody. But still, small biobank in Japan has claimed to the big biobank because in Japan there is big national bio, 6 national biobanks, maybe as you know, and also the very big mega bank in Tohoku University. I know that all of the less than 10 biobanks get the big fund from government, and very small money are distributed to the small one. Of course, the small biobanks hesitate to support to the thinking of the Japanese government. So and then recently they changed the mind. First, they make databases to share, of course, in English, everything. But I'm sorry, I cannot show this one because there is not yet open to the public. But the more detail, the online catalog for biospecimen was almost established in Japan. Maybe in near future, open to the public. So very detailed. Also, as you explained to do the very detailed information, check the list of their characteristics was there. Yes, maybe in near future. I think I already checked and also closed databases. I hope. I think I agree. Completely agree with you.
So as the director of biobank, so how often do you need to or how do you keep updating with all the standards that are coming up, or the international standards that
coming up, because normally 2 years or 3 years, and every year we should evaluate, right?
Yeah.
I guess so, something like that.
Yes.
Of course, our director decide. But sometimes, of course, it takes money. You talk about right? Yes. Yes, I cannot say that, but in the standardization, the biobanking, ISO's biobanking, maybe the government is not directly fund to us, but on the other hand, the medical field, almost same one, ISO 1587. And almost the accredited, right, parallel one, the laboratory accreditation is 17025, and this one is 20387, and medical one is medical testing accreditation standard is 1587, something like. It was recently the government make regulation. Regulation means that if we start to this kind of work, the medical check, so that they should get this ISO standard. So and then government is supposed to continue the standardization funding. Right? The Japanese system like that.
So please give a warm welcome to Dr. Jung-Sook Lee. Please.
Good morning. Thank you for nice introduction for me. Yeah, and thank you for inviting me and giving me the opportunity to talk here. Yeah, thank you. I would like to talk about the standardization of the case testing as I focus on case testing. I'd like to show my experience to obtain the ISO 9001. And first, I'd like to introduce the CREEP and case testing briefly. That CREEP is the largest government-funded research institute for biotechnology research in Korea. As Korea is under the Ministry of Science and ICT. As our mission, Korea's mission is to carry out the R&D activity in bioscience and biotechnology, and to support the infrastructure, provide the infrastructures to support the research of bioscience. So we have so many infrastructure, the centers related to such as the microbes and some plant or something else. This is the KSC buildings, new buildings. The KSC is the Korean Collection for Thai Cultures, is moved to current status 3 years ago. So the main campus of KRIP is in Daejeon, middle part of the Korea. But we have the two branch institute. One is the northern part of the Ochang, and the second is the other part of the Cheongno in the Jeonbuk branch. We moved to Jeonbuk branch. Here is in the January, so we did at the time we had very big snow. During the 1 month that we moved from main campus to these locations. We used this building. Second and third floor is the focused management of microorganisms and the related researches. The first floor is the administrative and the auditorium, and the basement is storage facility. KST's function is 3. We have the first one, the collection and the preservation of biological resources, and their distribution to industries, academic, and institute. A second is develop the core technology for valuable bioresources. It means we isolated some valuable microorganisms. One, the identification is in the report the new novel strains, and when we developed the cultivative or the preservative method according to each microorganism. The last one is the construction of local and international networks of biological resources and their related information. So we have some relations in the domestic, in the small collections, or the collections under the other ministry. And then we also together the discussion of something else that like the ACM is the member of the Asian members. KSBC, history of KSBC is started from the 1985. Our institute KRIP is originally under the KIST. The KIST is a kind mother of by Institute of Korea. So in 1985, we obtained the official approval from the Ministry of Science and Technology as a Senbank node. So that all the researchers called the KSBC as a Senbank now. And at the time that we became a member of the WFCC, or the Federation of Cultural Collections, and after next year, we became a member of the WDCM, or the Data Center for Microorganisms. And in 1990, we designated the International Depository Authority (IDA) from the WIPO, or the Intellectual Property Organization, based on the Budapest Treaty. And 2004, we obtained ISO 901 certificate for quality management of the system for the management of biological resources. And it is the special system is the 2008 we designated as the national repository of bio-research product, we called just the bioproduct. It means that Korea has some regulations. The researcher have to the bioproduct from the project funded by the already funded by the government have to deposit to the national repository. The CRIP is one of the national repository related to the biological resources. So from since 2008, we have managed the deposit in these bioproducts and preserve and distribute the other Korean researchers, and then they if the distributor studies with the byproduct and then have some special hit, they got the hit, they can share the benefit with the original depository. So it is the focus. And then the 2016 we moved to Temple branch. KCC is a director, general curator, and some the system, and then 7 curators, and we handle the most of the microorganisms, bacteria, ectomycetes, anaerobes, archaea, extremophile, and with including the microalgae, filamental fungi, and histone. And we also small part of the animal cell lines also. And then we have a large number of we manage the large number of the plant cell lines also. And then we have an idea, I said before, so that we manage the virus and genetic materials of some embryos and the plant seed also have to handle with the patent strain. And then some we manage some this like the biological resources under the byproduct materials. We have some preservation facility. One is the lyophilized samples for the preservation room. The second is the decontamination tank. The third one is the freezers room. And the bigger facility we will have in the main campus in Daejeon. This one shows the number of the holding at HTC. Until last year, so we have more than 34,000 strains. The main part is gram-negative bacteria, and we have also some patent strains and more. This slide shows positive cases in 2018. In 2018, about 2,000 strains have been deposited and cases from the researchers, the domestic and the abroad. You can see. Last year's case, gram-negative is also our first part. And last year we got the many parts of many strains of the mold from the domestic researchers. This slide shows number of cases strains reported in International Taxonomy Journal. You can see the IJC or something else is reported. There are novel strains, and about more than 300 species strains are reported in International Taxonomy Journal. So until now, KCC has holding the more than 3,000 type strains now. But this slide shows the distribution of, the 2018. You can see some decrease. We have some same problems, the distribution of strains. Last year, that we have distributed more than 6,000 strains. Now the Korea has some regulation according to each ministry. So we apply to quarantine and the animal disease, some the human-related issues, something else. We now it's controlled by the regulations. So it is delayed some the distribution, or some researchers have to check what kind of the regulation. And you know the CBD and our protocol, we have checked all kind of the issues. So maybe the problem is related to that, I think. Most widely distributed strain is the gram-negative bacteria TBRC, and last year we distributed so many anaerobics. Nowadays, many researchers interested in the microbiome, that especially gum microbiome. So nowadays, many researchers orders requested anaerobic bacteria. So this is increasing the anaerobic bacteria, and you know the lactic acid bacteria is a probiotics, so also the widely distributed. Yeah, you can see we distribute the strains, company, the institute, industry, university, institute, and so on. And then we also distribute the strains outside, so the abroad, the increases, you can see. And then many researchers publish the documents, and the number of journal referencing of the case strain is increasing. So usually the more than 400 case strains referencing the journal of the world. And from now on, I talk about ISO. Kawasaki, Dr. Kawasaki mentioned already. So I briefly the ISO 901, and then to share the information that my experience to obtain the ISO 9001 of the case this year. You can say ISO 9000 family addresses the various aspects of the quality management and contains of some of ISO best known standards. The standards provide guidance and tools for companies and organizations who want to ensure that their products and services consistently meet customers' requirements and that quality is consistently improved. ISO 9000 family required these items. The first is scope. The scope talk about the standards, how it apply to organizations. Next one is normative references. References, you can see from the Kawasaki-san. So it gives some the ISO, other ISO management systems. And then the terms and definition. Terms and definitions gives the definition used in the standard. And then the general requirement. General requirements gives ISO requirements for the overall quality management system. The next one is management responsibility. Gives ISO requirement for the management law in the quality management system. And then resource management. And product utilization. The next one, measurement, analysis and improvement. The next one, continual improvement. These are the requirements for ISO 9000. KCC obtained the ISO certificate 2004. At that time, it's the first time in Korea to obtain the field of biological resources center. So it is certificate, and we have the manual, including regulation and some procedures. This quality manuals contains 11 regulations and 12 procedures at the time. And first one is I'd like to the regulations. We have 11 regulations for the management of the biological resources, the quality management system, then record management, document management, education and training, internal examination, identification traceability, and corrective and preventive action, non-conforming biological resources management, equipment, the media and reagent management, and last one is computer program management. It means the handling of the databases. Usually most of the cultural collections are constructed in the database online. So we control the online database and offline the data sheet. This one. Each one is you show first the quality management system operation has list of the contents, scope of application, purpose, definition of terms. What is the quality management system? Quality management system documents, including manual, regulation, procedure, and so on. And then responsibility of the authority. Who the responsibility authority that including for the director or curator and management procedure basis and the relationship to quality policy and the quality management system, and then management documents, related documents, and review and evaluation of quality management systems and the report management, and then last one related documents. Quality record is also got the same contents, scope of application, definition, responsibility. In that case, it's the curator and the person in charge. And procedure, identification, collection of quality record, building, search, storage, protection, quality, the intention and deposition, something else, and the record management. And quality control, quality document, scope of application, responsibility, in the case of the director and curator, procedures. Education and training, responsibility director, curator, internal lecturer qualification, type of education, internal training, the external education, the procedure, education means it's very different. Education, internal training, external training, training for new employees, it was included, and management educational history and evaluation report of the result, and eligibility, and so on. The internal examinations, you can see that the term and definition of terms you can see include quality inspection, objective evidence, non-conforming, and auditor, responsibility of authority for director, auditing leader, the auditor, the curator as the examinee, and higher examinations, regular examination, and the temporary examination, auditory qualification, and procedures like this. The next one is identification of traceability management. You can see responsibility of authority for director and curator, and identification history, identification numbering, assessment, assignment method, so traceability. Corrective and preventive actions, you can see. Non-conforming biological resource management. The definition of the terms, non-conforming product. What is non-conforming product? Special, return, disposal. And the responsibility of authority for curators, criteria of non-conforming products, and procedure of the handling of non-conforming products. And equipment management, responsibility and authority for the director and curator, the person in charge of the handling of the equipment, each equipment. Equipment history card, equipment management procedures. And we use the main kind of the media ingredient in the cultural collections, so the responsibility and the authority and procedure that handling the media and ingredient. The last one is the computer programs directly create. Many of the cultural collections may construct the database, the own databases, using the computer programs. We also have the main database, the online database, and we updated on something else. We checked it and traceability to the code. And from now on, I briefly procedures, the 12 procedures. First four is related to case strains. It means case strain means the focused on the just non-commercial research only strain. The second was the patent strain, handling of patent strain. The last one is the handling of bioproduct. It is these strains are used by commercial, but they have when they have some hit or a special issue, they have to share the benefit with the original depositor. It is the common form of the documents of the procedures. First one is the deposit biological resources of KCT strains. This part is the names. I'm sorry, as a Korean, but we have no English version. So the name and then the enactment date and the revision date, something else, input and output. In case of this, the input is deposit strain, output is deposit, and the deposit procedure, and some storage of documents and what the user. It is the chart, flow chart for the procedures for the deposit biological resources, and this side is the personnel, the person in charge of this, and this is the contents of the each flow chart, each procedure, and this part is responsibility and then right, and then is related documents numbers, and then we have some document files, the followed of the procedures. So we usually, when in case of deposit of biological resources, we receive the strain with application form. We check the application form and the strains. Nowadays we have to get the more and more checklist that because of the CBD and Nagoya protocol and the regulation of the pathogenic strains related to plant quarantine or animal disease, and something related to human pathogenicity. So we check all kind of the issues. We especially now the Nagoya protocol check that it is well isolated this strain, well isolated the state. In case of some non-party of the Nagoya protocol, researchers deposited their strains. There is no problem to not apply to the Nagoya protocol. But some countries is have strict laws related to the Nagoya protocol. In case of the India, they have the law since 2002, I know. So the adoption of the NAWA protocol has been adopted in 2014 maybe, 2014, but the Indian researchers, they are trained, applied to their national laws since 2002. We have checked the related documents or related the certificate check, and then check the deposit strain, contamination or something else, and then check the viability and the purity. Usually, in case of the bacteria, we check the 16S RNA sequencing, and they compare the sequence with the sequence get from the original depositor. And then after confirmed all kind of the checklist, and then we assign the accession numbers and the preservation by the step. And then finally we send the issue, the certificate. This slide shows presentation, preservation of the biological resources. The procedure form is the same contents, but then we receive the strain data bank and then cultivate on the condition. Usually, according to the condition by the original depositor, or when we if we get the strains got from the other culture collections of Swiss, and then we according to the instructions. And then we check biological purity and then preservation. But you know, according to bacteria or something, the microorganisms, we have so many kind of the preservation method. The basic method is freezing or lyophilized, but in ingredient for the prospective reagent is either change it according to the bacterial algae and one giant yeast. So we check the issue and then made the preservation reagent, preservation solutions, and then preserve it. We usually separate the three blooms. One is the lyophilized and pool loop, and the second is liquid nitrogen, and third one is the deep freezer. And then we also two of them deposited in the mega facility in the main campus. This is the distribution of biological resources. So you can receive the order. We usually confirm the payment and then check the MTA. Then it is an important point related to our protocol. So we prepare the distribution and send the strain by the EMS or something else. The last one is claim. We receive the claim and then review the claim by the curator, each curator, and then check the strain, cultivate the strain, and then we identify that strain, and then confirm the strain finally by the curator. Then usually we resend the strain if we have we did not find the special problem. So we usually resend the strain. From now on, we related to the strain. It is another issue. We received the strain with the application form. It is the domestic deposit or international deposit between a little different related documents. So we received the strain with the document application form. We checked all kind of the document and confirm the payment. Yeah, and it is different point is the patent patent deposit. Usually, for the patent depositor have to pay the payment, the preservation, and check the deposit strain. It is a little different. In case of case strain, we check all kind of the identification for the 16S RNA sequencing, 18S, or the ITS or something else. But according to patent law, we just check the viability test. We check the viability and the purity, and then we take a photo and then assign the accession number and the preservation, and then issue the certificate. And distribution of patent strain, it is a little different. Received the order. In case of the patent strain, one is the order from the original depositor, and second is the third person. So the person get the permission from the original depositor or get the permission from the patent office. Most of the researchers can get the order, the patent strains, if they have permission from the original depositor of a patent officer. So that we check the document related to this, and then we receive the component the payment and they prepare the distribution. It is different from the case strain. In case of the patent strain, we usually check the viability before the distribution of the strain. So we check the purity and the viability, and we take a photo, and then we got the certificate, and then the prepare distribution. Usually we send the strain as the active purchase in case of the patent strains. And certificate of viability patent strain. Some depositors request the viability certification of viability. In that case, we will receive the order, and then confirm the payment and check the viability and take a photo, and then we issue the certificate. In case of the patent strain, if the strain did not grow, like it did not have the viability, the original depositor have to re-deposit the strains according to the patent laws. And then some depositor want to change the name. So usually first the depositor deposited their strains as the example of the Ashlos SP. But and then they gave the name, the exact name, and then they have to got the new certificate for the change the name. So in that case, confirm the payment, and then we change the name in the database and in related sheet, the strain sheet, and issue the certificate. From the remaining four procedures is related to the final product. So the procedure is the same, similar to the case strains, but some checklist is different. Receive the strain with the application form. In that case, we have to check the project name, a project leader, project number assigned to the government. We check, and then we added all kind of data in the bioproduct database and check the deposited strain and viability and purity, and then assign the numbers, the preservation, and finally issue the certification. And preservation is almost same. You cultivate the strain on the condition, given by the original depositor, and check viability and purity, and the purity check with the molecular assay or something else, and the preservation. Distribution of byproduct. In the case of the order is the payment is a little cheaper than case strains. And then in the case of the most of the researchers request the bulk time of the hundred of the strains or something else that we can prepare the distribution. Usually the active cultures or in some cases it is lifeless samples, and that's some of the strains. In the case of the bioproduct, we receive the claim, so we review the claim and check the strain that come from the strain is the active strains. And finally, I will show the table comparing ISO 20387 Kawasaki and ISO 9001 in KCC, and also the best practice guidelines, you know. So this accreditation ISO new accreditation ISO documents are based on the ISO 9001 and also the best practice guidelines. So 5 types of the requirements are assigned to each document entries. You can see. So the ISO 20387 is based on the existing ISO 9001 and also the best practice guidelines. So it is believed the BRC, especially the microorganisms, which already has ISO 9001, can proceed without the difficulty in receiving ISO 20387. Now that in Korea, so we also check the ISO new accreditation ISO 20387, and then we discussed the Korean Korean standardization types. Maybe there's this year maybe the set up the Korean styles. Yeah, thank you for your attention.
Thank you very much, Dr. Jung-Sook Lee, for the detailed information about the handling and management of bioresources in Korea, including common strains for research, type strain, patent strains, and bioproducts. So I would like to welcome comments and questions from the audience.
Thank you very much. I have one question. You say that you distribute a large amount of strain to outside, inter-country and in-country.
Yeah.
But I would like to know that the Korean, you have the many products.
Yes.
Did you have the calculation for the how many percent the proportion of your strain or something to make a business? Yeah, that proportion have you have data or not?
Yes. Most of the cases strains are distributed in the domestic. Yeah, and some abroad outside the researchers, I think including the major is China. I think Chinese researchers deposit their strains to KCC and then want to get the strains from the KCC, and USA and other country also tested some strains nowadays, and we also exchange the type strains between the other project collections. We usually the MBRC HHM in Japan, and we exchange some European countries, this in Germany, LMG and the Belgium, and we also exchange the strains between the CIP in France, something else, and some the Thai researchers also deposit their strains to KCC, and they some of them is distributed to Thai. But I'm not sure the exact percent of the more than 70% is the domestic to the domestic. The other is the exchange over the outside maybe.
Have deposits a lot of patent stain for animal
Yeah.
as well. Can you elaborate more what kind of the biological resources this derived from animal?
Animals, yeah. And in Korea, 4 IDAs, we have 4 IDAs. One is that especially of the focused on the animal cell lines. The human cancer-related cell lines. So many researchers deposited the cancer-related cell lines deposited in the culture collections related to just the cell line bank. Okay? And the others are deposited in our collection, but it is not much percent of the total. So we get usually the main part of the deposited patent strain is the microorganisms. The animal cell lines, the small part of the patent strain.